Okay, today we're covering the lab 2, covering chemistry. I was going to say the chemistry subject. So let me share my screen so I can get into the document, and then we'll work on filling that in. Then we'll go back and look at some of the images and relate some of this content to those images. And I'll briefly show you where some other notes are that are covering bond types. Okay, so excuse me. The first exercise, we're looking at different solutions, assigning a value, determining or assigning whether they are acid or alkaline, and the relative hydrogen concentration gradient. Now, I'll just preface this by saying, when it comes to quiz or exam questions, I'm not going to ask you what is the pH of gastric juice or urine or bleach or blood. Well, blood maybe. Blood is going to come back over and over again, 7.35 to 7.45. It will generally be this, a specimen or a fluid has a pH value of such and such. I'll give it to you. And you'll determine whether it's acid or alkaline and what the hydrogen ion concentration is. Okay, so as you know, on the pH scale, 0 to 14, 7 is smack dab in the middle, which is neutral. From 7 to 14 is considered alkaline, and from 7 to 0 or 1 is considered an acid. If we are alkaline, we're going to consider the hydrogen ion concentration low, and if it's an acid, it's a high concentration, and neutral will just be neutral. Now we know the farther away we get from neutral, the stronger the acid and the stronger the base or alkaline fluid is. But we're just trying to say, look, is hydrogen ion concentration increasing or decreasing, from that point at neutral. Okay, so gastric juice, let's say 3.5. We'll make, because it has a lot of hydrochloric acid, which is about 2. So we'll say 3.5. Is it acid or alkaline? It's acid. And the hydrogen ion concentration would be high. Then we go to urine. Hey, how are you? There's my dog. I'm zooming a lab right now, and the dog's now dreaming, and I can hear his feet going, on my office floor. Okay, urine is generally a little on the acidic side, let's say about 6.5. So it too would be an acid, and the hydrogen ion concentration would be high. Bleach is around 11. That would be an alkaline solution, and the hydrogen ion concentration would be low. Blood pH is 7.35 to 7.45, and that would be slightly alkaline, so the hydrogen ion concentration would be low. And distilled water, pH value of 7, it's neutral and neutral. Okay, then we're moving on to atoms and elements. We're going to identify the symbols and some of the model colors that go with each symbol. This is helpful to understand what each element is when it comes to looking at the organic molecules and models that we have images for. All right, so the symbol for carbon is a capital C, and the model color is black. Right? Then we have hydrogen with a capital H. Its model color is red. Oxygen is capital O, its model color is white. Nitrogen, the symbol is capital N, its model color is blue. Sodium is capital N, lowercase a. Chlorine is capital C, lowercase l. And we'll identify specifically what the colors are, because one is purple and one is green, and I can never remember. So we will get to that when we look at the images. But we just call them, when we see sodium chloride together like that, it's a salt. But one purple, one green, we call it Barty's balls. So we'll look at that shortly. Okay, and then when it comes to chemical structures and reactions, we're going to identify the building block of each of these families of molecules, and and perhaps the intermediate molecules. Right, so let me just kind of describe what we're looking at here. Right, we have on the top headings, we have carbohydrates, proteins, lipids, and nucleic acids. Those are our 4 categories of organic molecules in the body. All right. So something that would be helpful is if you practice writing this out, which I recommend doing is buying a whiteboard and doing this, is color code your families. So the carbohydrates might all be in green, proteins with blue, lipids in red, let's say, and the nucleic acids in orange or something, or black. So you identify them that way. Right. Going across, right, now we have on the left-hand side building blocks of each of those, the intermediate molecules of each of those, if there are some, right? Because lipids and nucleic acids, we don't have any. And the macromolecule of each, which we do have of each category. All right, so let's take carbohydrates, the building blocks of carbohydrates. Oh, and by the way, the bold terms, bold printed terms to the top here are what go into these blanks. I filled out most of it for you. All right, so let's go to building blocks, carbohydrates. Those would be monosaccharides. Mono means one and saccharide means sugar. And they are glucose, fructose, and galactose. Right, then if we stay in that category, we move down to the intermediate molecules, then we have disaccharides, 2 sugars, sucrose, maltose, lactose. And then the macromolecule would be a polysaccharide. Examples are glycogen and starch. Glycogen is the storage form in in animals. Starch is the storage form in plants. All right, then we go to the protein category. All right, so the building blocks of proteins are amino acids. We put a couple together and we get a dipeptide. And we put multiples of them together to form a polypeptide. An example of that is albumin. We have other examples as well in the subsequent exercise. All right, then we have fats, lipids. All right, so on one side, that first column, that is for the macromolecule of the triglyceride. Right? So we have these 2 terms that are going to go into here for the building blocks. There is no particular intermediate molecule, and then the macromolecule is a triglyceride. It's a neutral fat. So the building blocks of a lipid up on top would be 1 glycerol and 3 fatty acids. All right, those are the building blocks of a triglyceride. Just to the right of that, right, we have our macromolecule called a phospholipid, and we'll look at that a little bit more in next week's lab when we look at cells and the plasma membranes of them. But a phospholipid is kind of a called modified triglyceride. A lot of it is the same as a triglyceride. We're going to swap out one part, which makes it have bond types that are mixed. One is going to be polar covalently bonded, one is non-polar covalently bonded, and thus it interacts with water differently. And a phospholipid, is the type of molecule we find in a 2-layer orientation that forms plasma membranes and surrounds most organelles in a cell. All right, so back to the top, the building blocks would be 1 glycerol, 2 fatty acids, and 1 phosphate group, a phosphate group. So very similar to a triglyceride, instead of having 3 fatty acids, we're going to take out a fatty acid, swap it out for a phosphate group, and that makes it a phospholipid. All right, then our last category are nucleic acids. The building blocks are nucleotides. There is no particular intermediate molecule, and our macromolecules are DNA and RNA. Okay, so down below, there are 2 types of reactions you need to be fluent with. We either have a reaction that takes smaller things and puts them together called dehydration synthesis. All right, dehydration means to remove water and to put together. So below dehydration synthesis, we have building block, and then an arrow pointing to a macromolecule. So remember, in chemistry, the arrow, written out in some kind of chemical formula always points to the product, right? So the things that are to, the other end of the arrow, I guess you could say, would be your constituents of the product, right? So we may have multiple building blocks that come together to form a macromolecule. Right, so that kind of reaction where you start with something small, you end up something larger because you're putting them together in lengths. It's called dehydration synthesis. Are bonds created or broken? They're created. And is energy stored or released? It's stored. Another question that should be in here, and by the time you have this lab, I'm going to add this in here, is water added or released? Is it removed or released? And if we dehydrate something, we're removing the equivalent of one water molecule. I knew there was one more thing I wanted to change in this lab, and that was it. So is water added or removed? It is removed. That's why we call it dehydration. We remove the equivalent of oxygen from an OH group from one molecule and a hydrogen from the other molecule, which is the equivalent of water. And that opens up a bond space between those building blocks, or a building block and maybe an intermediate molecule, or multiple intermediate molecules to put them together to form a macromolecule, perhaps like a polypeptide. All right, so dehydration synthesis. Bonds are created, energy is stored, and water is removed. Now we can do the opposite. Hydrolysis means that we start with a larger molecule and we end up with something smaller. So maybe a macromolecule, and now the arrow's pointing to the left to the building block. And when we get to the images, I'm going to give you an example because I show you some pictures of molecules. If I start with this one and I end with this one, what kind of reaction is it? All right, so we ask, are bonds created or broken? Well, here we're broken because hydrolysis means to add water and lysis is to break apart. Is energy stored or released? It's released. Is water added or released? Water's added. Okay, then we move on to part 4. Using both models and different samples, which means maybe bottles that have labels on them that say that they are a sucrose molecule, or it's albumin, or it's cooking oil, that it makes it a fat. So we're going to apply, what types of bond types and functions are to these different molecules, whether they're in a model form, like a ball and stick kind of a model, or labeled boxes or bottles of things. And again, this will become a little more clear when we get to the images. Okay, so we're going to start over here on the left-hand side. A molecule, a sodium chloride. The bond type, you're going to identify that, and I give you the functions. So the bond type up on top is either ionic, polar covalent, non-polar covalent, or hydrogen. All right. So the bond type of sodium chloride is ionic. Then we go to water, one water molecule, one H2O. The bond type is polar covalent. Now, the term here, we're going to ask you a different type of question. If I have more than one water molecule, more than one polar covalently bonded structure, it could be water and water and water, or it could be two waters, or it could be a water and a glucose molecule or amino acid, right? Two polar covalently bonded things. What is the attractive force between those two molecules to each other? Not within each molecule, but from molecule to molecule. In this case, one water molecule to another water molecule. And that would be a hydrogen bond. Functionally, let me just go back up to the top. Sodium chloride forms ions in solutions. They can carry electric charges for nerve and muscle function. That's good. One water molecule, there is no particular function because you're never going to have just one. And multiple water molecules dissolves and transports most polar covalent and ionic molecules because like dissolves like when it comes to solvency. Okay, monosaccharides, glue-frugal, right? One sugar, single sugars, polar covalent bonds. Functions of quick energy used to produce ATP, the building block of a carbohydrate. We said that the intermediate products, we are not going to have to know any particular function for, so don't worry about those. Polysaccharide, glycogen or starch, right? That is, the bond type is polar covalent, and it functions as short-term energy storage in the liver and the muscles in animals like us, or, starch is to form energy storage in plants. All right, amino acid, polar covalent bonds, it's the building blocks of proteins. Polypeptides, or large proteins. Really, polypeptide is really the better term for it because the whole category is protein. Right? Or on the bottom, albumin specifically, both are polar covalently bonded. Now, when it comes to generally polypeptides, right, they make all the complex molecules in your body that have sophisticated functions. So they form things like bones, hair, muscle, antibodies, most hormones, enzymes. Anything that has very unique, complicated structure or function is going to be a protein, because fats just store energy for a longer period of time, and cushion inside organs, and the carbohydrate family is just a quick energy source versus kind of a short-term energy, or a quick energy source versus short-term energy storage. It's all just about using it for energy somehow, some way. But proteins, they form all kinds of things in the body, and they can be unique, like antibodies, and enzymes that bind to particular molecules. Okay, albumin, we'll get to know this over and over again, especially in AMP2. It maintains an osmotic gradient in plasma, which is the fluid part of blood. All right, so proteins, all these proteins, polar covalently bonded. Okay, then we're moving on to a glycerol and fatty acids, non-polar covalently bonded. It's the first non-polar building block of lipids, and a triglyceride, non-polar covalently bonded, functions as long-term energy storage, insulation, and cushioning. Then we go to the phospholipid. We said that was a modified triglyceride, right? The bond type now has two different types of bond types. It has areas that are polar covalently bonded and non-polar covalently bonded. Right? And its function is a cell membrane component. So it's going to be a building block of a cell membrane. We'll see that more next week. Right? The phosphate group is polar covalently bonded. It likes water, so we say that it's hydrophilic. And the glycerol and 2 fatty acids is non-polar covalently bonded and said to be hydrophobic, or its behavior with water is hydrophobic, doesn't like it. Okay, nucleotides, polar covalently bonded, building blocks of nucleic acids, DNA and RNA. Right? These are the macromolecules of our nucleic acids. Right? Both are polar covalently bonded. In the center of DNA, there is hydrogen bonding that occurs there. So maybe center of DNA, you can write hydrogen bonds, and they carry genetic instructions. DNA stays in the nucleus. RNA becomes kind of a photocopy of the DNA and can leave the nucleus and go out to organelles like the ribosomes and be read and synthesize amino acids, or take amino acids and make a polypeptide. We'll look at that more in next week's lab. We just right now kind of want to know the basics of them, but I will go back and say, I'm going to mention here about proteins, because proteins is unique things about them. Proteins have a lot of hydrogen bonds within the large molecules, I will tell you. And because of that, it has a lot of hydrogen bonds that are subject to denaturation. So let me just back up again and just say proteins, unique structure. For them to have these unique functions, they have what's called a primary, secondary, tertiary, and quaternary structure. And they're the only family of organic molecules that does that. The primary structure is the initial amino acid sequence out of the 20 amino acids. And let's say you have 100 amino acid chain, any difference in any amino acid in that chain doesn't make it entirely a different protein, but it gives it different functional capabilities as a protein. So let's say you had 20 different colored beads in a box, and you could string, whatever sequence of the 20 colored beads that you have on a string of 100 beads. And if you swap out just one at the 10th position or the 99th position or the 56th, and you swap out, let's say, a yellow bead for a blue bead, is that it will change the way that amino acid functions compared to the original version. And when you look at 100 to 200 amino acids in a sequence, when you can simply change an amino acid here and there along that long chain, and that's the only thing you change, it starts to give you a sense of how many different proteins there must be in the body. All right, so then that's its initial sequence. When it goes from the initial sequence and you put it in a spiral or a beta pleat, what keeps it in that shape is the positive negative of one amino acid being attracted to the positive and negative of the other amino acid. And this has to do with the bond types. So this is why you need to know polar covalently bonding, or what's happening with a polar covalently bond. So when the negative end of one amino acid becomes approximated to the positive end of another amino acid farther down the chain, they kind of come together like magnets, and they keep this shape. And as that long amino acid full of little amino acids, or the amino acid chain full of amino acids have positive and negative ends to them because they are polar covalently bonded, is that they keep this spiral shape or a pleated type of a shape. Those are hydrogen bonds from one amino acid to another. Then you go to the tertiary stage, which is take a long, maybe helix, and you tie it in a knot like a pretzel. That's a tertiary stage. And again, you maintain that shape for the same reasons you maintain the secondary shape to it, is because positive and negative ends of other exposed amino acids become attracted to each other, and now they keep it in the form of, let's say, a knot or a pretzel. The quaternary structure may be multiple pretzels all coming together, and again, exposed positive and negative ends of exposed amino acids become attracted to those in another adjacent pretzel or knot. And so that is its quaternary structure. And those positive and negative attractions like that are the hydrogen bonds. We're going to talk about it briefly in a PowerPoint if your instructor hasn't discussed polar covalent bonds versus non-polar covalent bonds. I have a quick PowerPoint in there that can go over anything that you're going to really need to know. Okay, so using part A, right, identify the primary or dominant bond type of each of your categories. So carbohydrates would be polar covalent. Proteins would be polar covalent. Nucleic acids would be polar covalent. Lipids are non-polar covalent. Right, so lipids are the only one that has an incompatible bond type with water, and water is the primary solvent of the body. And if water is polar covalently bonded, it makes sense that other molecules in the body are going to be polar covalently bonded as well. All right, but not everything is, and thus it forms nice walls and borders between one area of a cell and another, or one area of the body and another, because water is going to be repelled by these fat molecules. Think of it this way: what happens when you put oil in water? Let's say you have a pot of water to go into make some pasta in or something, and you put oil in it. Oil doesn't dissolve, right? It beads up until you put enough heat there where it really agitates and breaks some bonds, but oil and water don't mix. Because they have incompatible bond types. Lipids, fats are non-polar covalently bonded. Water is polar covalently bonded. All right, so what is denaturation? Denaturation is the breaking of hydrogen bonds. What two variables cause denaturation? High temperature and extremes of pH, most commonly low pH acids. And once you start to denature those proteins start to unravel from their quaternary to the tertiary structure to their secondary structure. And as often there's no going back. So, once those proteins start to break apart, they're never going to be able to perform the functions that they needed to, and you need them because you need those for survival. Okay, then we go to solubility, and here we're using water as our solvent. You have solutes and solvents. Solvent is the medium you're going to put solute in. And generally, the solvent is generally something liquidy. In this case, it's going to be water, polar covalently bonded. And the solvent is the substance you're going to put in there. And try to get it to be suspended throughout your solvent. So we are going to look at each of these molecules. We're going to use relative molecule sizes, either small, medium, or large. Bond type is either ionic, polar covalent, or non-polar covalent. And then we're going to describe the solubility, high, medium, or none. And I'll just tell you that small things are easily soluble, as long as you have compatible bond types. Large molecules with compatible bond types will have medium solubility. And then there's only going to be one thing that has no solubility, and you can probably guess what it is, right? So we're talking about the solute is water. All right, so sodium chloride, molecular size, small, bond type is ionic, and its solubility is high. I mean, how easily does salt dissolve in water? Pretty easily, especially if you just stir it. Okay, glucose. It's relatively small as well because it is a building block. Bond type is polar covalent and its solubility is high. Starch, its molecular size is large. All right, is large. The bond type is polar covalent and its solubility is medium. And the reason why is because it's just starches are long chains of things. So yes, it has compatible bond types, right? But it doesn't, but it's awfully large to suspend. It takes more, it's more difficult to suspend. So we'll say it's at a medium solubility. Oil, its molecular size is large as well. It's a non-polar covalent bond. So what would you expect its solubility to be? None. And then we have albumin or protein. It has a large molecular size. Right? Its bond type is polar covalent, and its solubility is medium. Okay, so now we're going to move on to the couple questions based on your experimental data. Which properties, size, bond type, or polarity, did the molecules with the highest solubility have in common? So they had the same size, they were small, and they had complementary bond types. They were polar covalently bonded, just like water. Which properties were different between the high solubility and the medium solubility? Well, it was, what was different, not what they had in common, was simply just size. They were both polar covalently bonded, but because they're larger, they considered to have medium solubility. In which molecule was insoluble and why? That would be the lipids, or the oil. Why? Because they were not only large, most importantly, they had an incompatible bond type. They were non-polar covalently bonded, while water is polar covalently bonded. Okay, and let me go back up to glucose here. If your particular instructor said to call that medium, that's fine too. It's technically very small because sucrose, maltose, lactose are very small molecules. But if your instructor said, hey, sodium chloride is really small and glucose, yeah, it's medium, that's fine. The size at that point doesn't really matter. It still has a very high solubility. As you know, you put in glucose or even sucrose, which is a disaccharide, you know how quickly it dissolves in coffee or tea. Okay, so that's where I'm gonna stop my share, and I'm gonna change now back to the course page. Hope y'all are still with me here. And let me go back over here. Okay, let me go back to my share. Go into the course page, which it's not letting me do. So let me, there. Now it should let me in there. You gotta kind of toggle back a little bit with zooming here. Okay, there it is. Now it pops up so I can share that other page. Okay, so now I'm going back to, now that we filled in that page, and sorry you guys, I've got a cold right now, so I'm a little nasally, I'm a little more nasal than normal. I've always sounded nasally, but I'm a little more congested than normal, so I apologize. And so hopefully I went through that. It wasn't terribly eloquent, but let me go to these chemistry bonds and I'll show you where I am. In lab 2 chemistry, we have, the lab, the document we just, filled out for the week. We had images below that, but I also put up here a little PowerPoint on chemistry bonds, in the event that you don't have me for lecture. and your teacher didn't go into a lot of detail, I can go ahead and open this up really quick. And I think the share will follow me, right? Is that you can always go through this PowerPoint if I went too quickly, right? And you can identify fairly quickly what these different bonds are. It's loading up right now, so just give it a moment. And I'll kind of zippy on through it. It's spinning, 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 so I'm just gotta be patient. Not my forte. Well, it says you may need to download it, so let's just do that. All right, I'm gonna stop my share and I'm gonna reshare because I got an idea. It won't follow me that far. So let me go back and reshare. Hasn't linked it up yet. There it is. Okay, so for those of you who want to have a little more background about chemical bonds, all right, we can just chemical bonds, forces to hold atoms of a molecule, or a compound together. We have the ionic, covalent, and hydrogen of the covalents. We have polar covalent and non-polar covalent. All right, goes through ionic bonds. It's an attractive force that holds ions together, formed when one atom loses and the other gains an electron, like a sodium and a chloride. All right, the positively charged ion that lost the electron is the cation. The other that gained the electron is the anion. You can kind of zip through this. Covalent bonds, we can either be non-polar, or polar. So it occurs when atoms share electrons, right? Non-polar covalent bonding means equal sharing of electrons. That occurs in lipids and lipid-soluble molecules like cholesterols and things like that. Polar covalent molecules, unequal sharing of the electrons. Water, carbohydrates, proteins, nucleic acids, any of the building blocks, our macromolecules or intermediate products found there. And then hydrogen bonds, attraction between molecules with polar covalent bonds, between water molecules and in the center of DNA, right? It could be a water molecule and something else polar covalently bonded, like an amino acid, like a carbohydrate. Hydrogen bonds are also found in long chains of proteins. And that's why hopefully you got into this a little more in lecture, because, you have what's called intramolecular hydrogen bonds and intermolecular hydrogen bonds. And that's the difference between through the center of DNA versus that found maintaining your secondary, tertiary, and quaternary structure of proteins. And then, hydrogen bonds, you can read through this. I went into a good amount of detail in the event chemical reactions. If I start with A plus B and the arrow points to C, then A plus B would be the reactants and C would be the product. If the arrow was pointing from C over to A plus B, then C would be the, kind of the reactants and A plus B would be the products, or the reactant, and the A plus B would be the products. So the arrow points to the product. This is just showing you that, that you could have a reversible reaction. Forms of energy, potential energy, kinetic energy, activation energy, catalysts, what they do, types of reactions, anabolic reactions versus catabolic. Anabolic reactions mean to build things up, to take small things and end up with a larger thing. Dehydration synthesis creates bonds, store energy, removes water. Hydrolysis breaks bonds, release energy, add water. Solutions and electrolytes, again, pH and so forth. So I'm going to stop that share. So feel free to go in there and look around at that material. It may be, quite helpful for you. Okay. Let me now go and break my share and reshare to the course page. So let me close that one out and go back to this page, to chemistry, and we'll go to look at the pictures now. Okay, so here we have images. Now that should be better. There we go, we're cooking oil now. All right, so let's look at some of these images. All right, here is the first image. So here we have what we consider to be props, so to speak, bottles that might be labeled with something like sodium bicarbonate in the back, or Rolaids like Tums that are antacids, baking soda here. You know, these are things that tell you clearly what they are. Okay, they act as buffers, they function to maintain pH and homeostasis. I don't ask you about buffers, but it gives you a good example of things that have labels on them, that props that you might need to know. All right, and then we have, so here he has like a picture of something that contains like a fluid that is gastric juice. This is that first chart that we filled out, you know, what might be the pH and what's the concentration of hydrogen ions. The pH is 4, 3.5, 3. It doesn't matter, you know, as long as it's in the acid range still. So I'm not going to ask you to memorize the pH, you know, the gastric juice at 3 or 4 pH, but you do need to know that if it's a pH value of 4 or anything less than 7, it's an acid and the hydrogen ion concentration is high. And that goes for any of these fluids, right? Bleach. It's not important you know the exact value, you know, but that you know that it's about 10 or 11. It's a base or alkaline, low hydrogen ion concentration. Distilled water would be 7, and the hydrogen ion concentration would be neutral. Pardon me. Okay. Now here we have our atoms. We didn't have urine. Now that I look at that, there wasn't anything for urine, I don't think. Go with about 6 to 6.5. Not that you can't have alkaline urine, because you can. People who have or vegetarians that have high vegetable diet, you know, they often have more alkaline urine than the average person. Okay, so here are the balls and sticks I was talking about. The balls and the different colors represent the elements. Hydrogen is white, right? That would be capital H. Chlorine is green, so I can never remember which is which. Capital C, lowercase l. Oxygen is red, capital O. Sodium is capital N, lowercase a. Purple balls, carbon is a black ball, capital C. Nitrogen, capital N is blue. And just check, I'm pretty sure I said that the hydrogens were white. And if I made a mistake, I'm very sorry. Hydrogen, did I say hydrogen was red or white before? Well, see, I have a cold. Right. Hydrogen is white, oxygen is red. I'm not going to ever ask you what, you know, I'm going to point to a red ball and ask you what element that is. I'll never do it, you know, but this will help you make better sense of the models that we're going to look at that are organic, you know, ball and stick models, we call them, representing monosaccharides or disaccharides. Okay, this one purple, one green, those are the Barney's balls I was talking about. This would be a sodium chloride. Don't call it a salt, although it falls in the salt category, because salts, you know, are technically a category of molecules that are formed when you have an acid and base interact with one another, right? And one of the byproducts is a salt. And that's where generally these will often come from, right? So we call it sodium chloride because those are the two elements there, and the bond type would be ionic. And then you should know a function associated with it from that, you know, from the chart. Forms electrolytes in solution, can conduct electrical impulses, you know, in nerves and in muscle tissue. Here we have a water molecule. We have 2 hydrogens and a red, right? As an example of polar covalent bonds. And we said one water molecule, we didn't have to know any particular function associated with it. Here it says just polar, but do give me polar covalent. Entitled, you know, these are some images from another instructor and they're great, but it's not necessarily the standard for the course. You always want to go with, you know, these are generally, virtually most of us are using these, you know, these images, but you need to go with whatever was in your lab exercises that week. Okay, now we're looking at 2 water molecules. So now this is that that third line on that chart and functions. What is the bond between those molecules? Right, so we have one polar covalently bonded water molecule and another polar covalently bonded molecule. All right, so the attractive force between them, like the negative end of the red to the positive end of the white hydrogen, that's a hydrogen bond between these two molecules. Or if I gave you 4 other molecules here, right, the force between each of these polar covalently bonded molecules, maybe even if I throw in an amino acid, it would still be hydrogen bonds. And they are subject to those 2 variables. Right? High temperature extremes of pH, especially acids. Okay, so now we're going into carbohydrates. And here we have one ball and stick model of a monosaccharide. You're not going to have to identify whether it's a glucose, a fructose, or a galactose. It's just a monosaccharide at this point. Now, if I gave you a bottle that said glucose, then you have to know that it was glucose. It was a monosaccharide. We'll do more of that in AMP2. All right, so you need to know the molecule, the bond type, and the function. It is a monosaccharide. It's polar covalent. X is quick energy, the building block of a carbohydrate. So if we put 2 of those together and link them together, and you have 2, what I call bracelets or handcuffs here, then what again is the molecule, the bond type, and the function. Right, so this would be a disaccharide, 2 sugars, polar covalent bonds, and no particular function that you have to know. All right, then we get into polysaccharides. You can't quite see this, but you see at least 1, 2, and off to the left, the model turns a little bit, so you see it sideways, but those are 3 monosaccharides linked together as a polysaccharide. Right? And then the box behind it says starch, so that's a polysaccharide. All right, so molecule, bond, and function. Right, it's carbohydrate, polysaccharide, polar covalent. Short-term energy storage. In plants, that would be starch. In humans, it would be, or in animals altogether, it would be glycogen. Okay, now we're looking at, oh, look at that. It's a bottle of oil. All right. So if we're looking at oil, we're in fats. All right. What is, it is a lipid, right? It is a triglyceride. Bond type is nonpolar covalent, and it's long-term energy storage. I'll say it's not just energy, it's long-term because it's much harder to go to those fat reserves and use it for energy. Right? It helps cushion and insulate organs. Right? Non-polar covalently bonded. It's a macromolecule too. Right. Here is an example of a fatty acid. I'm just going to tell you. Unfortunately, we don't have a picture here of the whole triglyceride in ball and stick. It looks like an octopus, or a tripod, I guess you could say, because you have 3 fatty acids all connecting to this 3-carbon glycerol backbone. This is a fatty acid. When you see a linear sequence like this of black balls, through the center, which are your carbons and hydrogens attached to them, the white balls, and you see that little double bond where you see that little circular circle of plastic straw there with the red oxygen. This is a fatty acid. So 3 of these come together to bind to a glycerol to form a triglyceride. So this is a fatty acid. It's a building block of a triglyceride. It's nonpolar covalently bonded. And you don't have to know an individual function for it. It's a building block of a triglyceride. That's fine. All right, here is the 3-carbon glycerol background of a triglyceride. So you have 3 black balls. This is glycerol. So 1 glycerol, 3 fatty acids forms a triglyceride. Right? It's the building block of a triglyceride, nonpolar covalently bonded. No function other than it's a building block of a triglyceride. All right, now we get into blue balls. All right, so proteins are the only organic family that contains a nitrogen molecule, and it's what's called the amine group. And the nitrogen is blue. Right? So this is the only family that'll have a blue ball and then a little double bond here over to the lower right-hand side of that molecule, and then the red oxygen and hydrogen off the end. So that one little unit, that's an amino acid. So you see one blue ball, one double bond, it's a building block, right, of a protein. Right? So it's amino acid, polar covalently bonded, it's a building block. We put two of those together, so a blue ball, double bond, blue ball, double bond. Now we have two amino acids, bound together. That would be a dipeptide. Right? Polar covalently bonded, no function. I'm kind of zipping through this now, you guys. And here we have three. Right? So blue ball, blue ball, blue ball, double bond, double bond, double bond. Right? So this represents a polypeptide. Right? Multiple blue balls, double bonds all along, right? Polar covalently bonded. They form enzymes, hair, nails, antibodies, blah, blah, blah. If we had, albumin, if you saw a bottle of just albumin, we want you to know the particular function for it, but otherwise, proteins, these are the functions, forms hair, nails, enzymes, antibodies, bone, things like that. If we saw the bottle of albumin, it maintains an osmotic gradient. That would be the specific function. Okay, this is kind of sideways, but this represents a double helix, all right, and that would be DNA. All right. The bond type is polar covalent, its building blocks are nucleotides, and it contains genetic material. If we have a single helix, which sometimes we can get a good picture of, it's a little hard to see because you have one side of the spiral, right? That would be RNA. It's another macromolecule of the nucleic acid family, right? And it's polar covalently bonded and contains genetic material. At this point, that's fine. Right? DNA, RNA, macromolecules in nucleic acids. Right. This is an example of a nucleic acid. Kind of looks like an earring more than a handcuff or a bracelet like a monosaccharide does. And it has, a variety of like brown and yellow balls that the other molecules didn't. All right, so this is a nucleotide. It's the building block of DNA and RNA, polar covalently bonded. Okay, so now we're going to show you how we're going to apply that hydrolysis, dehydration synthesis. And we said that arrows, point to our product. But we're also, I would generally say, look, if I start with A, which A is indicating a dipeptide because you see the two blue balls there, and I end with B, which is multiple blue balls, that would be a polypeptide. So I start with something medium size, I end with something larger, okay? So if I started with A, and I have to tell you this, I started with A, I ended with B. What kind of reaction was that? It would be dehydration synthesis. If I started with A and ended with B, or multiple A's and ended with B, did I break bonds or create bonds? Well, I created bonds. I started with took some small things and put them together to make one big thing. If I started with A and ended with B, did I store energy or release energy? And I stored energy because whenever you create bonds, you store energy in those bonds. All right. Okay, dehydration synthesis. But there are other questions that could be applied to that. That's why you're not limited to them. Okay, now we're going to give you a different question. It's like, okay, if I start with A, which is the polypeptide, and I end with B, which is a dipeptide, you can't quite see the rest of it, but it's a dipeptide. Or even if it was a monopeptide, I mean, an amino acid, it doesn't matter. All right, so if I start with something big and I end up with something smaller, if I start with A and I end with B, what kind of reaction? Hydrolysis. If I start with A and I end with B, do we store energy or release energy? Well, we release energy because we break bonds and we release the energy. Did I add water or remove water? I added water. think that's the last picture, hydrolysis, and then anything that goes along with hydrolysis. Bonds broken, energy stored, water added or released. Okay, that's the end of the material. Let me think if I have any other thoughts to share with you on that. I think that's really it. Most of my questions, they come right from those exercises. So please, now that you have one quiz under your belt, is that if you haven't already done so, you print off the document there, these lab exercises we're going to fill out, or download it, and then fill in all those blanks, because that's where I get my questions from. And although I'm kind of covering it with you right now, is that you want it all filled in so you can just look at the 2, 3 pages, 4 pages rather quickly and study from it. And then exam questions are very similar to the lab questions. There's just more of them, that's all. Okay, then I'm going to break the share here and end the Zoom. Thank you very much.